But before that, let's have a brief introduction. Nowadays, infectious diseases is the main cause of mortality and morbidity worldwide. The different types of infection caused by pathogens including bacteria, fungi, viruses, and parasites are ever increasing. Old age, diseases such as cancer and immunosuppressions, and others are some contributing factors. Although the field of diagnostic microbiology has substantially evolved over the past few decades, it is still heavily reliant on cultures and serology, which, while being cost-effective, is largely time-consuming and sometimes less sensitive and specific. Molecular biology, on the other hand, has developed with rapid and confirmatory diagnosis. Molecular methods have progressed beyond identification to detect antibiotic marker genes, fastidious bacteria, and uncultivable microbes. This is the reason why we came up with the applications of molecular diagnostics in microbiology. So, what is molecular diagnostics? Molecular diagnostics is the collection of techniques that is used for the detection of genomic variants, aiming to facilitate detection, diagnosis, subclassification, prognosis, and monitoring response to therapy. It is the rapidly developing area of laboratory medicine that investigates human, viral, microbial genomes and the products they encode. The question is, how molecular diagnostics in microbiology works? Every organism contains specific DNA sequences, so the molecular diagnostic makes the specific DNA visible. It allows rapid detection by matching of nucleic acid sequences, by detection of viral antigens released during the infection process, or by the detection of increased antibody presence with specificity for viral proteins. So, let's move on to the applications of molecular diagnostics in microbiology. First, classification of organisms based on genetic relatedness or genotyping, identification of microorganisms grown in culture, early detection of pathogens in clinical specimens, rapid detection of antibiotic resistance, detection of mutation, detection of toxigenic from non-toxigenic strain, and lastly, used in forensic medicine. So, let us recall of what is genotyping. Genotyping determines differences in a genetic complement by comparing a DNA sequence to that of another sample or a reference sequence. It identifies small variations in genetic sequence within populations, such as single nucleotide polymorphisms or called as SNPs. Genotype determination can also be used for the identification of susceptibility and prognosis for any disease and to find out for a specific treatment, thus leading the way towards personalized medicine. Second, identification of microorganisms grown in culture. Many organisms occupy a diverse range of habitats, and so the only way of knowing the origins of a particular contaminant is to identify or characterize it with a specific strain and then match it with a strain elsewhere in the environment. Knowing how to identify bacteria or other unknown microorganisms that you have encountered can help you assess where a clone came from. The third application of molecular diagnostics contributes to the development of a disease. Now, with the help of molecular diagnostics, the detection of toxigenic from non-toxigenic strain is very possible and is less time-consuming. Lastly, it is used in forensic medicine. Forensic molecular biology emphasizes molecular techniques such as DNA profiling, repetitive DNA in human genome, and many more. So, those are the applications of molecular diagnostics in microbiology. And lastly, let's enumerate the benefits of molecular diagnostics. First, can create a new cancer screening tools. Second, can inform designs of new treatments. Can monitor treatment effectiveness. And lastly, can predict patients' response to treatment. So, to sum up, molecular diagnostics is the outcome of the fruitful interplay among laboratory medicine, genomics knowledge, and technology in the field of molecular genetics, especially with significant discoveries in the field of molecular genomic technologies. The application of molecular diagnostics in microbiology provides for the qualitative and quantitative detection of pathogenic organisms in patient specimens, microbial identity,